Okay guys, it's Miss Lynham again. We're going to do another recording. This time our recording is going to be a little bit of basic organic chemistry with an introduction to the biomolecules. Okay, so if you have your notes, we should be on this slide that starts with organic chem, the study of carbon. So just kind of want to show you a minute the organic chemistry part, and some of y'all might be organic chemistry lovers and might be organic chemistry majors in college whenever you go there. If you take organic chem, you're really studying a lot of carbon compounds because it is the study of carbon-based compounds. Organic is the term that we use to term anything that contains carbon. Although carbon dioxide is an exception to that, it's a waste product, it's a gas that does contain carbon, but carbon dioxide is not organic. But lots of other compounds that do have carbon are organic, and we're going to talk about quite a few of them in this unit. So the first thing I want to do is show you the amazing ability of carbon. And if you remember from last unit, I gave you carbon, the little formula off the periodic table, and it has the atomic mass of 12, and it has the atomic number of 6. So if you remember back to Bohr modeling, when we talked about drawing the Bohr model for that, the nucleus, my little pen is not working very good, the nucleus is going to have its 6 protons, okay, 6 P's and 6 N's. Okay, hopefully you could do that really quickly. And then we have to plot 6 electrons. So in the first orbital, we plot our 2 electrons. We've got 1 here, we have 1 here, and then the second orbital, remember we can have a maximum of 8, but we only have 6 because it's carbon. 2 are plotted, so we have 4 left. So 1 goes here, 1 here, 1 here, and 1 here. So, when you look at the Bohr model, what kind of bond do you think carbon is going to be more apt to form with other things? Hopefully you said your answer is covalent. Okay, so the answer is covalent bonding. And because of that, carbon is found in all of our biomolecules because they're all going to involve covalent bonding. Okay, so if you look at the outer valence shell, the valence electrons, there's a need to have 4 more to be stable. Okay, so carbon needs 1 here, needs 1 over here, needs 1 here, and needs 1 here. So it's more likely to be sharing its valence electrons with other things. Just some of the elements that you're going to see carbon sharing with: sulfur, phosphorus, oxygen, nitrogen, hydrogen, and actually carbon is going to be forming bonds with other carbons. So last unit we introduced these sponge elements. Carbon has a tendency to share its outer electrons with these other elements. Okay, so you're going to see it written a lot of times. We will write it where you've got the carbon like this, and we're going to have the lines coming off, and that just shows you that carbon has the ability to form 4 bonds, 4 covalent bonds with other atoms. Okay? So that's just kind of your intro. So you can see in the picture here, here are a few examples of structural formulas of things according to, let me get the little movie working, here we go. You can see some structural formulas. So these carbon-based compounds come in a variety of sizes, lengths, shapes, and their appearances. So you're going to see them, and they look very, very different. Like this one, the linear structure has all single bonds between the carbons. This one over here has single bonds between the carbons, but you've got a branched chain, okay, where you've got a carbon branching off of another carbon. Every single carbon still has its 4 valence electrons being filled. If you look around every carbon, you can see the 4 covalent lines. So this one is okay, and this one is okay, but what you see in these first 2, if you were to add up all the carbons and the hydrogens, you would see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogens, and 1, 2, 3, 4, 5 carbons in this one. And if you do the same thing over here, you will indeed find 5 carbons and 12 hydrogens. So these 2 structures that you see here are known as isomers to each other. Okay, don't confuse that with the word isotope. An isomer is something, a compound, that has the same chemical formula. This is the chemical formula, C5H12, but there's a different arrangement of atoms whenever you look at the structural formula. So that's a structural isomer. Okay, in biochem or in organic chem, there are other types of isomers, like the second example that you can see in Part B, where you've got not a single bond between two carbons, but a double bond. Now remember, each carbon can have 4 covalent bonds. So if this carbon uses 2 of its electrons to bond with another carbon, there are only 2 other ones left over. So that's why you see this line coming off and this line coming off, and same thing over here. So whenever you throw that double bond between them, okay, this one is showing you there's an X, which is like a variable group on this side, and then a hydrogen, and then a variable group on this side with a hydrogen. Well, this one has the same formula as this one over here, okay, but they are known as cis-trans isomers because of the location of that variable, that X group. So the 2 X's are on the same side, it's a cis isomer. If they're on opposite sides, it's known as a trans isomer. So it's just another way of looking at things in organic chemistry that can have the same chemical formula but different structures. And the third one that you see down here, this one's kind of really interesting. It's called an enantiomer. An enantiomer is one that is like a mirror image, is how we like to describe it. It's kind of like you see this person's hand. If you take the thumb of this hand and line it up with the thumb of this hand, so it would be like you clapping your hands together. The thumb and the thumb meet up, the first finger and the first finger meet up, and so forth, but yet the thumb is on opposite sides according to the picture. This is like a 3-dimensional ball and stick model. Okay, so these are L&D isomers. They also have the same chemical formula, but they're mirror images of one another. Mirror image meaning if you take this H on the thumb, line it up with this thumb over here, the outside, that would line up there. This right here, the CH3, would be like the palm of the hand, would line up with the palm. The top lines up, and then of course you've got the NH2 and NH2, which would represent pinkies. So if these were hands and you clasp them together, those would be enantiomers. Okay, so that's just examples of some different types of ways of looking at these organic compounds, these organic molecules. I should say molecules actually, because molecule indicates covalent bonding, which is what carbon does. Okay, the last thing on this slide is this term hydrocarbon. Hydro, remember, in our cases we've talked about water with this, but it's the H part, the hydrogen component. So hydrocarbons, which are really important in fossil fuels, you may have heard of these before, if you've read any kind of, read anything, any research on fossil fuels. Hydrocarbons are compounds that are composed only of carbon and hydrogen. So these 2 up here would be hydrocarbons at the top because they're C5H12. They are very hydrophobic. There's nothing about them that makes them polar, so these fossil fuels tend to be scared of water, fearing water, non-polar. Okay, so that's a little bit of intro about the carbon and the importance. So now we're going to talk about these things called functional groups. Okay, so functional groups are things that you can add. They're groups that you can add to a carbon chain, a carbon skeleton, that can give it different properties. Okay, so for example, if you look at this first one, if I took off this OH group, you would just have C's and H's. That would look like a hydrocarbon, which would be very nonpolar, hydrophobic. Well, in organic chemistry, sometimes there are these things called functional groups that you can add to a carbon chain that make it act very different in the case of this first one. The first one that you see here is called the hydroxyl group. Okay? Hydroxyl is written as OH. You'll usually see it like this. Occasionally you'll see it as in the form of HO, but normally it's the O that is written like it's bonded covalently. This is the covalent bond line. The oxygen is covalently bonded to the carbon, and then the hydrogen is covalently bonded to the oxygen, but sometimes you'll see it written in this fashion as well. So this one's called hydroxyl. It's the OH group. And so in this first example, you can see that the carbon still has its 1, 2, 3, 4 valence electrons are filled, but it's sharing with an oxygen here with a hydrogen bonded to that. So this now becomes one group. This group right there is called the hydroxyl group. What that does to this carbon chain is it now makes it very polar. So the OH group is a polar group. We refer to these as alcohols. We're going to talk about one that's present with a phospholipid. It's actually called glycerol. So anytime you see OL on the end, the OL is the alcohol group. OL usually means that it's polar, that it's got a polar property because of that hydroxyl group. Okay, the second one, I like to say carbonyl, but some people say carbonyl. The carbonyl or carbonyl group has to be a carbon that is double bonded to an oxygen. okay? So you have to see this in the structure like this right here. That's a carbon double bonded to an oxygen. As far as how it's written, it's usually written with the covalent bond line to the carbon, and the carbon is bonded to the oxygen. So you don't have to draw the two lines here. It's just understood that the carbon has a double bond with an oxygen, but you'll see it written like this. Covalent bond CO, but the C is in a double bond with the O. Okay, now there's two ways of looking at this. Okay, this gets a little bit more complex. If the C double bonded to O is within the carbon chain, okay, so like here's the first carbon, this is the second carbon, here's the third one. So this is on the end over here, and this is on the end. This C double bonded to O is in the middle of that carbon chain. If it's in the middle or within it, we call that a ketone. Okay, so if that carbonyl or carbonyl group was within the carbon chain, it's known as a ketone. If it is at the end of the carbon skeleton, like this C double bonded to O is on the last carbon that's in here, it's known as an aldehyde. Okay, so that carbonyl group is at the end of the carbon skeleton. So one more time, the group is called carbonyl, okay, or carbonyl. It's a carbon double bonded to an oxygen. If it's within the carbon chain, it's a ketone. If it's at the end, it's known as an aldehyde. Okay? All right, third one, moving right along. There's quite a few of these little functional groups that give things different properties. The third one is known as carboxyl. This is one of my favorites because it sounds like a box car, like a train. But if you look at this, it looks like chew, C-H-O-O. So it's kind of like choo, choo, chugga, 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 chugga. Get it? Because it's a box car. So it's a chew group. Just kidding. All right, so this one is a C double bonded to O, but on the same carbon, it's single bonded to an oxygen and a hydrogen. Oh my goodness, y'all, this looks like, right here, this looks like a hydroxyl group, and this right here looks like a carbonyl group. So if the C double bonded to O, which looks like carbonyl, is on the same carbon that an OH is attached to, this is now a brand new functional group. You would not call this a carbonyl and a hydroxyl. This is now a brand new group called a carboxyl. Okay, this makes something, if you add that C double bonded to O with an OH to a carbon chain, it makes it act as an acid. Because what it's going to do is donate the H+, this breaks off, and it's going to donate that H as an H+ in solution. So if you have this C double bonded to an O with an OH on the same carbon, it's called a carboxyl group. They're known as carboxylic acids because they make something act like an acid because it donates its hydrogen ions. Okay? Make sense? All right, on to the next one. Make sure I didn't skip one. Yep, we're good. Okay, next one is the amino group. This one's really important in proteins. Okay, the amino group is going to be a nitrogen, okay, it's got to be a nitrogen attached to two hydrogens. So it's a nitrogen with a covalent bond to a hydrogen and another covalent bond to a hydrogen. So we call these things amines. And you sometimes see them written like this, with the covalent bond line to the nitrogen, and it's NH2 because there's two hydrogens. Okay, this makes something act like a base. Whoa. So instead of donating H+ ions, it's going to remove H+ ions from the solution, and when it does that, it becomes NH3. So it's going to take an H+ from a solution, add it to the two H's that are there, and becomes a different chemical. Okay, so that's the amino group. Okay, the next one, sulfhydryl. Just look at the name. Sulf is like sulfur. Hydryl is like hydrogen. So this is a covalent bond from the carbon chain to a sulfur, and when that is bonded to a hydrogen. These have the nickname or the being called thiols. These will also be really important in stabilizing protein structure. We're going to talk about these in like a day or so, because when proteins take on different shapes and they're in different structures, they can form these things called disulfide bridges between a sulfhydryl group of one amino acid and a sulfhydryl group of another amino acid. So in this case, what you're seeing is a sulfur bonded to a hydrogen. Okay, that's the sulfhydryl. All right. I believe these might be the last two. Woohoo! Okay, the next one is the phosphate group. This one's kind of easy because phosphate sounds like phosphorus, and you have to have a P. Okay, there's your P, your phosphorus. So you're going to have this phosphate group doesn't just have the P, it has 4 oxygens bonded to it as well. So you can see it written as covalent bond OPO3, or sometimes you'll see it written as PO4. You'll see it written like that instead of like this. But it's a phosphorus with 4 oxygens around the outside. These are known as organic phosphates. Okay, we'll definitely talk about these when we get to nucleic acids, because DNA and RNA, ATP, are going to have phosphate groups. And what these do is give the molecule a negative charge, gives it kind of a negative charge. As you can see this, look, negative, 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 and that's because of the electronegativity of oxygen. It tends to react with water and help in the release of energy. And like I said, we'll talk about these with ATP, because ATP has 3 phosphate groups, and we'll talk about how it does that when we get to ATP. So that's the PO3, bond OPO3, or PO4 group is the phosphate group. Okay, the last one is a methyl group. You guys probably have heard of methane, which is a lot of carbon and hydrogens to methane. Methane gas is one of those. But the methyl group is a CH3. Okay, so it's written covalent bond to the C is bonded to 3 hydrogens. This is definitely important whenever we talk about gene expression. You might want to write on here. When we talk about gene expression, which is the expression of genes with eukaryotic and prokaryotic organisms, we'll talk about that. The methyl group is going to be important because we'll talk about something like demethyl or methylation. Okay, so that's kind of an important one that we'll talk about as we get throughout the year when we go through our different groups. Okay, so why don't you try this one? See if you can point out some of the groups on this one. Okay, so just to show you a little bit, you should be able to take this and identify some of the groups present on here. Okay, so can y'all name this one right here? Hopefully you said phosphate group. Okay, how about this one right here, OH? That's hydroxyl. Okay, hydroxyl. Could you tell this one is a C double bonded to an O? This is not an OH, it's an H. So this is a what? Carbonyl. C-A-R-B-O-N-Y-L. That's a carbonyl or carbonyl phosphate group, and there's your hydroxyl group. So there's 3 functional groups added onto this one carbon skeleton. Okay, how about this? This is CH3. There's one. CH3. What are those, guys? Hopefully you said methyl. Those are your methyl groups. Okay, over here, hydroxyl. Okay, there's your hydroxyl. Oh, look, here's a C double bonded to an O, and it's within the carbon chain. What is that one? What did you guys pick for that one? Because then there's this one over here that's at the end. Which one's the aldone? That's good. Which one is the aldehyde and which one is the ketone? Can you guys tell? How'd you do? So this one, hopefully you got this. Ketone. Aldehyde. H-Y-D-E. My pen is not working. That's the aldehyde. Okay, could you also tell another phosphate group right there? Okay, you getting the hang of it? Okay, down here, how about this one? This one has a lot of these OHs. Hydroxyl, 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 hydroxyl. Look at all those. And then this is C double bonded to O. That's carbonyl or carbonyl. It's at the end, so it's also an aldehyde, like this one over here. Okay, how about over here? Here's an NH2. That's amino. Here's a C double bonded to an O, and then there's an OH. This right here is the carboxyl. C-A-R-B-O-X-Y-L. that's the carboxyl group. And then look at this bad boy, sulfhydryl. That's the sulfhydryl. Okay, so now see if you can go back once you're learning these, because it's a lot to learn all these functional groups. See if you can add in, like, which one made it called an alcohol, which one is the thiol, which one is like a carboxylic acid. You know what I'm saying? Like, see if you can add in the functions and what it does to the different functional groups. Make sense? Okay. Just for the intro, I'm going to do like two or three more slides and then we'll be done with this one because I'm going to work on proteins during class. But just so you can see the intro for these biomolecules, for the next, I would say week and a half, we're going to be focusing on what I call the big four biomolecules. Okay, so biomolecules, just look at the word. These are life molecules because bio means life. Molecule refers to the fact that they're going to involve covalent bonding. They're very stable substances, and they're going to be sharing their outer valence electrons with other things. Okay, the big four, if you remember, here they are: carbs, lipids, proteins, nucleic acids. Most are polymers. The reason it says most is because this lipid group right here is very unique. It's very diverse. So the lipid group does not fit into the same little like philosophy as the other macromolecules. I sometimes call them macromolecules or biomolecules. Macro refers to the fact that they are big polymers. Remember, poly means many, mer means subunit or unit. So these are going to be many units long. Carbs can definitely be many units long. Proteins, nucleic acids, many units long. Lipids may not be, like they're not all long, but we're going to have long fatty acid tails when we start talking about our saturated and unsaturated fats. So those big polymers, if they have them, they have the monomer is the one unit that makes up the polymer. Mono means one, mer means unit. Okay, so in this picture right here with these colorful little circles, this entire long chain would be the polymer, and then each one of these circles would be a monomer. So this polymer is made up of many monomer, different colored circles. Okay, the black line you see that I'm drawing red right here, that's the bond. That would be your covalent bond that is linking those monomers together. So this picture might represent, let's just say, starch. Starch is a carbohydrate that is really long, and it's made up of glucose plus glucose plus glucose plus glucose plus glucose plus glucose plus glucose, on and on and on. So all of those glucoses would be covalently bonded together to make a really long polymer of glucose monomers. Does that make sense? So in the picture down here that you can see, if I scroll it up some, there is your one glucose. Now this looks different than what I just showed you on the functional group pictures. This would be the ring structure of glucose. You saw it drawn out in linear structure, which is where you saw the carbon bonded to the carbon, bonded to the carbon like this. But you can also represent it in chemistry and organic chem drawn in a ring. And because glucose is a 6-carbon sugar, it's drawn out with a hexagon. Okay? So that's glucose. This one over here that you see is fructose. So if I join glucose to fructose, I get a disaccharide because it's two monomers linked together, and that gives it a whole new name, a whole new thing called sucrose, which is much different than the two things individually. So those two would be linked together. I'll show you the bond right here. That would be the covalent bond between this monomer glucose and this monomer fructose. So it's the same thing as this blue one linked to this orange one, like that through a covalent bond. Okay, this bottom one is showing you glucose plus glucose plus glucose plus glucose, which has a certain arrangement. So that's a longer polymer of a bunch of glucoses linked together. Okay, does that kind of make sense? All right, so the last few things I just wanted to go over: how to break a polymer and how to put a monomer together with another monomer to build a polymer. Okay, so in conceptual basis for this biochem, we're going to be breaking and building polymers and monomers the whole time. Okay, so if you look at this first slide that's regarding this, the first one is called dehydration synthesis. If you use your root words, guys, to synthesize means to make. If you're dehydrating, you're losing, I'm going to draw an arrow down, losing water. This literally translates as to make something and lose water as a result. It's sometimes given the name condensation reaction, okay, or condensation, because condensation is when you have water that forms on the outside of, let's say, a glass, So it's the same thing. If you lose water off that glass, it's condensing. It's a condensation reaction. This is in order to build. So let's change colors here. I want you to make sure that you circle or highlight or make note of this. Here's the word, is building. If we're building a longer polymer. That would be like taking a circle, linking it to a second circle. We just made a 2-circle unit, so we just built it longer. If I want to take a third circle and link it, I just built it and made it longer. So I'm going to build this polymer through dehydration synthesis. My bond will be covalent. My loss is water as a waste product. This does require an energy input, so we're going to have to use, when this happens in our body, we are going to have to use some ATP for energy for this to happen, and it's going to be facilitated and triggered by tons of enzymes. You have enzymes in your body that will help make this process happen a lot faster, okay? So in the diagram, if you look, if I've got, here's a monomer, here's my second monomer, here's my third monomer. If I want to link this fourth monomer right here to it, okay, remember, these are formulas. So this might be the formula right here that I'm circling the 3. It might be C6H12O6. Let's say it's a glucose. This might be C6H12O6. So there might be on this number 3, there's a hydrogen atom hanging off of that monomer. This one will have an OH hanging off of its monomer. When they come close to each other, they combine to form H1, H2O, there's my water that comes out, and now my result is I'm going to have a bond between them, which is this line right there, and now I just built a larger polymer. That's dehydration synthesis. Okay, you can probably already visualize what the opposite would be. Instead of dehydration synthesis, we're not going to make a bond, we're going to split a bond. It's called hydrolysis, it's called hydrolysis. Okay, but I like to break it up so you see the syllables. Hydro is water. Okay, so it's hydro, and then to lyse means to split or break. So this is water breaking or water splitting. This is going to have the opposite as far as water is concerned. We're going to need to add water into this reaction, this chemical reaction, in order to break the bond. So the covalent bond between our monomers is going to be broken. We're not going to need energy. We're going to release energy. And it will also be accelerated with enzymes. So both dehydration synthesis and hydrolysis are going to require enzymes. So for the final picture, here you go. We've got this this polymer made up of 4 monomers. Does that make sense? A polymer made up of 4 monomers. I'll erase this line right here. Okay, so here's my polymer. If I want to break, I'll change the color so you can see. If I want to break this one off, I'm going to add water into the chemical reaction. The water will split or dissociate into an OH and an H. The OH adds to this one monomer, the H adds to that one, and now I have a split right there between that. And so my result is I just split or broke a bond. Okay? And we will stop there and start with proteins in class. Great job.